Good morning, students. In the today's lecture, we are going to discuss the life cycle of the Agaricus. In the last lecture, we have discussed the life cycle of the Aspergillus, and the topic for the today's lecture it is the life cycle of the Agaricus. Here, the first of all, the systematic position or the classification of the Agaricus. We are here following the classification which was proposed by the Alexo Paulus and the Mims in the 1979. Okay, Agaricus, it is included in the kingdom Mycete, division Mastigomycota, subdivision Basidiomycotina, class Basidiomycetes, order Agaricales, family Agaricaceae, and the genus is the Agaricus. Okay, here the classification of the Agaricus, which was proposed by the Alexopoulos and the Mims in the 1979. Okay, as per this classification, all the fungi these are placed in a single kingdom, Mycete. Okay, here that's why the Agaricus it is placed in the kingdom Mycete, division Mastigomycota. Okay, Mastigomycota, that is the division. That means in this division, the fungi these are not producing the any type of the motile cell. Okay, that's why it is referred as the Mastigomycota. Then subdivision Basidiomycotina, class Basidiomycetes, because in the Agaricus the spores which are referred as the Basidiospores, that is these are produced during the reproduction, and that's why these are placed in the class Basidiomycetes. Okay, subdivision Basidiomycotina, class Basidiomycetes, order Agaricales, family Agaricaceae, and the genus is the Agaricus. Okay, that is the systematic position or the classification of the Agaricus as per the Alexopoulos and the Mims in the 1979. Okay, then regarding the habitat of the Agaricus. Agaricus, it is a edible fungus and it is commonly known as the mushroom. Okay, you all know regarding the mushroom. Okay, the edible fungus, and it is commonly known as the mushroom. Okay, that is just we have discussed in the introduction to the fungi, that is the study of fungi. It was previously started with the study of the mushrooms. Okay, because the botanists they were get attracted to the mushrooms due to their colors, and that's why the study of the fungi it is believed to be started with the study of the mushrooms. Okay, the Agaricus, that is the edible fungus and which is commonly known as the mushroom. In old literature, the mushroom it was known by the generic name Saliota. Okay, in older literature you can find references of these mushroom by the generic name Saliota. Okay, the mushrooms these are the saprophytic fungus which are found growing on the soil humus. Okay, saprophytic fungus means that is these are deriving the food material from the dead organic matter. Okay, and that's why these mushrooms these are found growing on the soil humus. These are found growing on the decaying litter on the forest floor. Okay, litter means that is the freshly fallen plant parts, that is it may be the leaves, or these may be the branches of the plants, flowers, fruits. Okay, all these which are fallen on the ground in the forest these are referred as the litter. Okay, and the mushrooms or the Agaricus these are found growing on these decaying litter on the forest floors. These are also found in the fields and the lawns, wood logs, and the manure piles. Okay, then these mushrooms these grows based in the moist and the shady places, and these are commonly seen during the rainy season. Okay, you all know that that is the mushrooms, that is they flourish very fast, especially during the rainy season. Okay, the mushrooms these grows based in the moist and the shady places, and these are commonly seen during the rainy season. Then these mushrooms or the Agaricus, these are cosmopolitan in distribution. Okay. Then about 17 species of the Agaricus have been reported from the India. These mushrooms, these are commonly known as the kukurmuta in Uttar Pradesh, and these are also known as the dhingri, or these are also known as the khum in the Punjab. Okay, mushrooms, these are popular as a kukurmuta in the Uttar Pradesh and dhingri in the Punjab. About 17 species of the agaricus or the mushroom, these are reported from the India. Okay, of these 17 species, Agaricus campestris, okay, it is popular, or it is commonly called as the field mushroom because it is widely growing, and that's why it is called as the field mushroom. And another species, that is the Agaricus bisporus. Okay, it is also known as the Agaricus brunescens, and it is commonly called as the white mushrooms. These are the common edible mushrooms. Okay, of these 17 species, Agaricus campestris, which is commonly called as the field mushroom, and the Agaricus bisporus, which is called as the white mushrooms, these are the common edible mushrooms. Of these two species, Agaricus bisporus, it is a cultivated mushroom. Okay, it is a cultivated mushroom, and it is widely cultivated for the food purposes. Okay, then some of the species of the Agaricus, these are poisonous. For example, Agaricus xanthoderma, and some species that may causes the gastrointestinal disturbances in some persons. For example, Agaricus placomyces and the Agaricus sylvaticus. Okay, here the Agaricus bisporus, it is the widely cultivated species of the Agaricus, especially for the edible purpose, or especially as a food material. And that's why here in this lecture we are going to discuss the description of the Agaricus bisporus. Okay, here we are going to discuss the vegetative structure of the Agaricus bisporus and the reproduction in the Agaricus bisporus. Okay, that is all about the habit and the habitat of the Agaricus. Then here, first of all, the structure of the Agaricus, it can be studied under the two heads. That is the first one, the vegetative mycelium, and the second one, the fruiting body. Okay, the structure of the mycelium. Here we are going to discuss the structure of the Agaricus into the two parts. The first part is the vegetative mycelium, and the another part, it is the fruiting body, which is commonly called as the Basidiocarp. Okay, here the first part of the structure, that is the vegetative mycelium. Here you can see the vegetative mycelium. The most important thing regarding the vegetative mycelium is that this vegetative mycelium remains underground, and it is of two types. Okay, vegetative mycelium, it is underground, and it is of the two types, the primary mycelium and the secondary mycelium. Vegetative mycelium, it remains underground, it remains buried in the soil, and it is of the two type. That is the first one, it is the it is called as the primary mycelium, and the another one, that is the secondary mycelium. Okay, the primary mycelium, it is formed by the germination of the basidispore. Okay, primary mycelium, here is the primary mycelium, and it is formed by the germination of the basidiospore. Okay, basidiospore, we will discuss how exactly the basidiospores are formed in the reproduction of the Agaricus. Just here, just remember that the primary mycelium of the Agaricus, it is developed from the germination of the basidiospore. It is hyaline, it is hyaline means it is a transparent, and here in this photograph you can see it is a septate. Since the transverse septum is present, or the partition walls are present, and it is monokaryotic. Monokaryotic, that means only the one type of nucleus it is present in the hyphae, or in the cells of the hyphae. And that's why here we are saying that the primary mycelium, it is hyaline, it is septate, and it is a monokaryotic. Okay, depending upon the strain of the basidiospore. Okay, basidiospore, it may be positive strained, or it is negative strained, and that's why depending upon the basidiospore, the primary mycelium, it may be positive or the negative strain. Okay, if the basidiospore it is positive strained, then it will germinate and give rise to the positive strained primary mycelium. And if the basidiospore it is negatively strained, then it will germinate and give rise to the negative strained primary mycelium. Okay, the primary mycelium it is short lived, and it soon get transformed into the dikaryotic secondary mycelium. Okay, here primary mycelium it is short lived, and it soon get transformed into the secondary mycelium. And here you can see the secondary mycelium it is dikaryotic. That means the each of the cells of the hyphae these are having the two different nuclei. Okay, two different nuclei. That is, the one nuclei it is positively strained nuclei, and another nuclei it is the negatively strained nuclei. And that's why this secondary mycelium it is said to be the dikaryotic. Why it is called as the dikaryotic? Because the two nuclei these are present in the each of the cells of the hypha, and these two nuclei these are of the opposite strain. One nuclei it is of the negative strain, and the one nuclei it is of opposite strain. Okay, the secondary mycelium it is formed by the fusion of the two primary mycelia of the opposite strain. Okay, two mycelia of the opposite strain, that is the positive primary mycelia and the negative primary mycelia, it get fused with each other, and then there will be the formation of the secondary mycelia. And that's why the cells of the hyphae of the secondary mycelium these are having the two nuclei. One is of the positive strain, and the another one it is of the negative strain. Okay, the secondary mycelium it is perennial, then it is branched, and it is septate. Okay, perennial, it is secondary mycelium it is perennial, it is branched, and septate. Then the hyphae of the secondary mycelium twist together to form the white hypal cords, and these white hypal cords these are known as the rhizomorph. RHIZOMORPHS. Okay, the secondary mycelium, that is the hyphae of the secondary mycelium, twist together to form the white hypal cords, and these hypal cords are called as the rhizomorph. And from these rhizomorph, here you can see the fruiting bodies are formed. Okay, and from these hypal cords, that is which are called as the rhizomorphs, from these rhizomorphs the fruiting bodies are formed which are aerial. Okay, here the primary mycelium and the secondary mycelium, that is these are completely buried in the soil, while these fruiting bodies these are aerial portion, that is which are growing above the soil surface. Okay, these are the fruiting bodies, and these are called as the Basidiocarp. Okay, the dikaryotic mycelium, it shows the centrifugal growth. That is, the hyphae grow outward from the center in the circle. Okay, here you can see there is the hyphae these are growing outward from the center in the circle. A circular colony of the hyphae it is thus formed in the soil. Circular colony of the hyphae it is formed in the soil, and at maturity the hyphae develops the fruiting bodies at their tips in more or less circular ring. Okay, and that's why that is these are also popular as the fairy rings. Okay, because these fruiting bodies these are developed in a circular manner, and these are called as the fairy rings based on the ancient belief that they marked the path of the dancing fairies. As per the ancient belief, that is these are marked the path of the dancing fairies, these are referred as the fairy rings. That is about the vegetative mycelium. And now the second part of the structure, that is the fruiting body or the basidiocarp. Okay, generally this fruiting body or the basidiocarp it is developed after the sexual reproduction in the Agaricus. But here we are discussing the structure of the fruiting body or the Basidiocarp, which is developed after the sexual reproduction. Okay, just we have discussed that is the two primary mycelia of the opposite strand, that is they get fused with each other, and then there will be the formation of the secondary mycelium, which is a dikaryotic, and this secondary mycelium will develop into the rhizomorph, and from this rhizomorph the fruiting body it is developed, and that fruiting body it is referred as the basidiocarp. Okay, here we are discussing the structure of the mature basidiocarp. The mature basidiocarp, it is an umbrella shaped structure. Okay, from this photograph it is clear that the structure of the basidiocarp, it is umbrella shaped structure with the long massive stipe. Okay, here the fruiting body basidiocarp, it is differentiated into the massive stipe. Okay, it is the stalk like region, massive stipe, and this structure it is called as the pilus. Okay, Pileus. Okay, that is here it is mentioned as a cat, but in the book it is referred as the pilus, Pileus. The stipe or the stalk like portion, it is thick, it is fleshy, it is cylindrical structure, then it is light pink or white in color. The umbrella like pilus or this cap region, it is about 5 to 12 centimeter in diameter. It is present at the distal end of the stipe. The upper convex surface of the pilus, here the upper convex surface of the pilus, it is white or it is light brown in color or yellow in color. Okay, that is the umbrella like structure, that is the cap or the pilus. It is about 5 to 12 centimeter in diameter. Then it is produced, it is present at the distal end of the stipe. The upper convex surface of the pilus, it is white colored or maybe light brown colored or yellow in colored. Then about 300 to 600 radially arranged gills, these are produced. Here about 300 to 600 radially arranged gills, these are produced from the inner surface of the pilus. Okay, here you can see the presence of the gills. Okay, the gills, that is about 300 to 600 gills, these are produced on the inner side of the pilus. All the gills, these are not of the same length. They may be of the full or half or the quarter length. The surface of the gill, it is enveloped by the fertile layer, and that is called as the hymenium. Okay, here the surface of the gill, it is covered by the fertile layer, and that fertile layer it is called as the hymenium. Hymenium. Okay, the fertile layer covering the gill, it is called as the hymenium. Hymenium. The gills, these are of the light pink color when young, but when they turn brown or purplish black at the maturity. Okay, these gills, these are the fertile part, because these are having the fertile layer which are called as the hymenium, and in these hymenium there is a presence of the basidiospores. Okay, and these gills, these are of the light pink color when young, but when they turn brown or purplish black at the maturity. Okay, that is all about the structure of the fruiting body or the basidiocarp in the agaricus, which is popularly called as the mushroom. Here the internal structure of the basidiocarp, that is the fruiting body. The basidiocarp, it is made up of the pseudoparenchymatous mass of the hyphae. Okay, it is made up of pseudoparenchymatous mass of the hyphae. Here the first of all, the transverse section of the style or the internal structure of the stipe. Okay, yeah, 1 minute. Here you can see the first one, that is the first photograph. Here you can see this is the transverse section of the pilus, this one A, and it is the transverse section of the stipe. The first of all, the internal structure of the stipe. The stipe, it is composed of the numerous longitudinally intertwined hyphae. Okay, here the stipe, it is made up of transverse section of the intertwined longitudinally arranged hyphae. The hyphae, these are compactly arranged and form the pseudoparenchymatous tissue in the peripheral region, whereas these are loosely arranged with the large intercellular spaces in the central region. Okay, that is, here you can see these hyphae, these are compactly arranged in the peripheral region, but in the central region here you can see these hyphae are loosely arranged, and there is a presence of the intercellular spaces in the center region. Okay, the peripheral region of the stipe, it is known as the cortex. Here you can see this is the peripheral region which is known as the cortex, while the central region where the intercellular spaces are present and where the hyphae, these are loosely arranged, this region, it is referred as the medulla. Okay, that is the internal structure of the stipe. Now the internal structure of the pilus, here this one is showing the internal structure of the pilus. The internal structure of the pilus, it is similar to that of the stipe. Okay, it is similar to that of the stipe, and the pilus, it is differentiated into the outer cortex. Here you can see it is the outer cortex and it is the medulla. Okay, the internal structure of the pilus, it is differentiated into the outer cortex and the central medulla. Okay, the cortical region, that is mm, it is made up of compactly arranged cells, and here this is the medulla region is loosely arranged, while in the center region here you can see the stipe region. Okay, we can see the stipe region which is present in the center. Okay, that is about the internal structure of the pilus. Now here the most important one, the fertile region, the internal structure of the gills. Okay, gills, these are very important part of the mushrooms, because this is the fertile region of the mushroom. Here you can see the internal structure of the gills. The internal structure of the gill, it is a complex, and it is differentiated into the three regions. Okay, it is a complex region and it is differentiated into the three regions. The first one, here you can see this is the trauma region, TRAMA. Then here you can see the second layer which is present, it is said to be the subhymenium. It is said to be the subhymenium and the hymenium. Okay, here you can see this is the hymenium. Okay, this elongated portion, basidium, and this one, this region, it is said to be the hymenium, and this one below, this one, it is said to be the subhymenium, and below the subhymenium there is a presence of the trauma region. Okay, thus the internal structure of the gills, it is differentiated into the three regions. That is the trauma region. Here this is the trauma region, then subhymenium, and this one, that is the hymenium. Okay, the first of all, regarding the trauma region, here the trauma region, this is the central sterile region of the gill, and it consists of many loosely arranged interwoven hyphae. Okay, here it is the central region of the gill, and here you can see it is consisting of the large number of the hyphae, but these hyphae, these are loosely arranged. Okay, this is the sterile region. Okay, it is referred as the trauma region. Then here the subhymenium region. Here you can see this is the subhymenium region. Subhymenium region, it is also referred as the hypothecium. Hypothecium. This region, it is situated on both sides of the trauma. Okay, this subhymenium region, it is situated on the both the sides of the trauma. Both sides of the trauma, it is formed by the lateral branches of the hyphae of the trauma region. The cells of these hypal branches, these are isodiametric. Okay, here you can see clearly the isodiametric cells which are present in the subhymenium. Subhymenium, it is made up of isodiametric 2 to 3 nucleate cells. Clearly in this one here you can see that is 2 to 3 nucleus. These are present in this cell. This is also the sterile zone like the trauma. Okay, both the trauma and the subhymenium, both these regions are sterile region. Okay, and then come here's the third region, which is called as the hymenium. Hymenium. Here you can see this is the hymenium region. Hymenium, this is the outermost layer of the gill. Okay, this is the outermost layer of the gill. It is fertile region and it is composed of hyphae of the subhymenium region. In this region the cells are arranged in the palisade like layer. These aseptate fertile cells, these are known as the basidia. Okay, here you can see the single basidium, and these large number of the cells are present. These are said to be the basidia. Okay, these are aseptate cells. Okay, these are palisade like cells. These are aseptate, there means the partition walls are entirely absent in these cells, and these are the fertile cells which are referred as the basidia. That is, these are club shaped. The basidia are present. On these basidium there is a presence of the spores. That is, in on each of the basidium, here you can see there is a presence of the four basidiospores. Okay, four basidiospores. Of these four basidiospores, two basidiospores, these are of the positive strained, and the remaining the two are negatively strained. Okay, remaining two are negatively strained. These basidiospores, these are developed after the sexual reproduction in the agaricus. Okay, after the sexual reproduction in the agaricus, these basidiospores are developed. Okay, that is all about the internal structure of the gills. Okay, just we have discussed the internal structure of the basidiocarp. In the basidiocarp, that is the fruiting body in the agaricus. We have discussed the internal structure of the stipe, then internal structure of the pilus, and also the internal structure of the gills. Okay, here the fruiting bodies basidiocarp, these are developed after the sexual reproduction. Okay, then here the reproduction in the agaricus, it takes place by the three methods: by vegetative reproduction, then asexual reproduction, and the asexual reproduction. Okay, reproduction in the agaricus, it takes place by the three methods: vegetative reproduction, asexual reproduction, and the sexual reproduction. The first of all, the vegetative reproduction in the agaricus. Vegetative reproduction in the agaricus, it takes place by the fragmentation. That is, these edible mushrooms or the agaricus, these are propagated by the vegetative means. Okay, just the small pieces of the dikaryotic secondary mycelium, these are used as the inoculum. Okay, if the small pieces or the fragments of these secondary dikaryotic mycelium, these are grown in the soil which is rich in the organic manure, then it will develop the fruiting body which is referred as the basidiocarp. Okay, normally these basidiocarp or the fruiting bodies, we refer them as the mushrooms. Okay, these fruiting bodies which appear to our eyes, which is the aerial portion which is growing on the soil, that is the fruiting body which we commonly call as the basidiocarp. Okay, here the vegetative structure, that is the primary mycelium and the secondary mycelium, these are underground portion. These are completely buried in the soil, but after the sexual reproduction, the secondary mycelium, it will develop the rhizomorph, and on the rhizomorph, then there will be the formation of the fruiting body which is called as the basidiocarp, and it is the aerial portion. And that fruiting body, that basidiocarp, which is commonly and popularly called as the mushroom. Okay, then here the asexual reproduction in the agaricus. Asexual reproduction in the agaricus, it is not the common method of propagation. Okay, it is not the common method of propagation in the agaricus. The asexual reproduction in the agaricus, it takes place by the chlamydio chlamydospore formation and the oidia. Okay, the chlamydospores, these are developed in the terminal or intercalary positions on the secondary mycelium. They germinate to produce the dikaryotic mycelium. Okay, chlamydospores, these are produced which are lateral or intercalary in positions, and on germination they will give rise to the hyphae, but generally these are developed on the secondary mycelium. On germination they give rise to the secondary mycelium, which is dikaryotic. Okay, while in some species the oidia, these are also formed, but these are involved mainly in the dikaryotization rather than developing directly into the new mycelium. Okay, that is all about the asexual reproduction in the agaricus. Okay, just remember that the asexual reproduction in the agaricus, it is the not a common method of propagation in the agaricus. Okay, the most common method of reproduction in the agaricus, it is the sexual reproduction. Okay, here the most of the species of the agaricus, these are heterothalic. Okay, but here the agaricus bisporus, that is the edible mushroom, it is homothalic. Okay, though the most of the species of the agaricus, these are heterothalic, but the edible mushroom agaricus bisporus, it is homothalic, but they do not have the sex organ in the traditional sense. Okay, that means the male sex organ and the female sex organs, that these are not developed in the agaricus. Okay, here the primary vegetative mycelium, which is formed by the germination of the basidiospores of the different strains, these are acting as a male and a female sex organs. Okay, here specialized sex organs are not developed, but the primary mycelium, this takes over the function of the sex organs. These primary mycelia, these are developed by the germination of the basidiospore. Okay, here the positively strained basidiospore will give rise to the positively strained primary mycelium, and the negatively strained basidiospore germinate and will give rise to the negatively strained primary mycelium. And here these two primary mycelia of the opposite strain will take over the function of the sex organs, and these two primary mycelia will get fused with each other, and then that is the such type of the sexual reproduction, it is called as the somatogamy. Okay, why it is called as the somatogamy? Because here the sex organs are never developed, but the two somatic hyphae, that is these are fused with each other, two somatic hyphae of the opposite strains, and they will get fused with each other, and that's why such type of sexual reproduction, it is referred as the somatogamy. Okay, this somatogamy, it will result in the diplodization and the formation of the secondary mycelium. Diplodization means that is the dikaryotic condition. It will develop the secondary mycelium, and the cells of the secondary mycelium, these are having the two oppositely strained nuclei. That condition which is called as the diplodization. After the fusion of the two primary mycelia of the opposite strain, there will be the development of the secondary mycelium, and this secondary mycelium then later develops the fruiting bodies, which are known as the basidiocarps. Okay, then the somatogamy between the two primary hypha of the opposite strain, it takes place through the three steps. The first one, plasmogamy; second one, karyogamy; and the third step, it is the meiosis. Okay, somatogamy in the agaricus, it takes place into the three steps. The first one is the plasmogamy. Plasmogamy, that is the fusion of just protoplasm of the two oppositely strained hyphae. Then karyogamy, that is the fusion of the two oppositely strained nuclei. And lastly, the meiosis, the diploid nucleus which is formed, it undergo the meiosis division. Here the first of all, how exactly the plasmogamy takes place. In this step, the two primary monokaryotic hyphae, here you can see the two primary mycelia which are monokaryotic and which are of opposite strain, that is they come in contact with each other. Then at the point of their contact, here you can see at the point of their contact the cell walls are dissolved and the dikaryon is developed. Dikaryon, that is the cell which is having the two nuclei of the opposite strain. Okay, that is the it is the positively strained nuclei, and this one is the negatively strained nuclei. Okay, thus a dikaryotic mycelium, it develops by the successive division of the dikaryotic cell. At the time of the division of the dikaryotic cell, both the nuclei of the dikaryon divide synchronously and form the four haploid daughter nuclei. Okay, here you can see that is these two nuclei, that is they does not get fused directly. Instead of this one, here you can see there is a formation of the specialized structure, and this specialized structure, here you can see the clamp connections. And due to the clamp connection, both these nuclei, instead of getting fused with each other, that is they will divide. And here you can see there is a formation of the four daughter nuclei, two of which, that is these are positively strained and the remaining two are the negatively strained. Okay, out of these two nuclei, that is one of positive and one of negative strain, that is these are transferred to the daughter cell. Here you can see that is how the daughter cell is developed, and in this daughter cell the one positively strained and the negatively strained nuclei, it is get transferred. Okay, and by the formation of the clamp connections. Okay, these specialized structures are clamp connection. Okay, that is then this dikaryotic mycelium, it is developed. Okay, this dikaryotic mycelium, it is developed, and the dikaryotic mycelium, it is a perennial, it is subterranean, and it produces the basidiocarps under the favorable conditions. Okay, these dikaryotic herpes, that is these positively strained nuclei and the negatively strained nuclei, that is they are not getting fused with each other. Okay, this secondary mycelium, instead it will develop the fruiting body which is called as the basidiocarp, which is aerial portion, which is commonly called as the mushroom. Okay, then how exactly these basidiocarps are developed after the plasmogamy? Here you can see that is the subterranean secondary mycelium. It takes the nutrients from the soil and then it forms the fruiting body or the basidiocarp. These basidiocarps develops as a small knot-like structures at the tips of the underground hyphae. Here you can see that is the underground is present, and on this underground hyphae there is a small knot-like structure. It is developed, which is called as the rhizomorph, and then these hypal knots enlarges gradually and give rise to the button stage. Okay, they will enlarges in the size and they will give rise to the button stage. Okay, and this button stage, then it will get developed into the fruiting body, that is it will get further differentiated into the stalk like region, which is called as the stipe, and that umbrella like region, which is called as the pilus. Okay, that is how actually the basidiocarps or the fruiting bodies, these are developed from the dikaryotic secondary mycelium. Okay, generally that is all you know, that in the dry season, that is the soil, it is very hard. Okay, and the buttons, they does not grow, and these buttons, they will remain underground. Okay, they will remain underground in the dry season when the soil is hard, but in the rainy season when the soil is moist, they grow rapidly and come out of the soil surface, and hence the large number of the basidiocarps or the fruiting bodies, these can be observed in the rainy season on the soil surface. Okay, just we have discussed that the secondary mycelium, it is perennial, that means it is living underground, and whenever the conditions are dry, whenever the soil it is very hard, that is these are present underground, and these are present underground. But when the conditions are favorable during the rainy season, when the soil becomes soft, then they will develop the fruiting body, which is referred as the basidiocarp and which is popularly called as the mushroom. Okay, that is how actually the basidiocarp or the fruiting body is developed in the rainy season. Okay, just we have discussed the up to the plasmogamy. That is how actually the dikaryotic mycelium it is developed, and from this secondary dikaryotic mycelium the fruiting body or the basidiocarp is developed. Okay, then the next step it is the karyogamy. Karyogamy, in this step, both the nuclei of the dikaryon fuse to form a diploid nucleus. Okay, here you can see that is the dikaryotic apical cell. Okay, here you can see that is the clamp connections are developed, then the nuclei, that is these are here you can see the tip cell. Now it is having the one positive strained nuclei and the one negatively strained nuclei, and here these two nuclei then they will get fused with each other, and there is a formation of a diploid nucleus, and that is how the karyogamy it takes place in the young basidium. Okay, that is these young basidia, these are present in the gill region. Okay, these are present in the gill region, that is these are present on the hymenium. Okay, this diploid nuclei then further undergo the meiosis. Okay, that is the third step of the sexual reproduction. The first one plasmogamy, second one karyogamy, and the third one that is the meiosis. Soon after the karyogamy, what happens that diploid nucleus it will undergo the meiosis division, and then the meiosis division which takes place in the young basidium, and there will be the formation of the four haploid nuclei. Okay, that means this young basidium it is now having the four haploid spores. Okay, it is four haploid spores, that is this out of these four haploid nuclei, that is two haploid nuclei, these are of the positive strain, and the remaining two nuclei, that is these are of the negative strain. Okay, remember that the diploid nucleus in the young basidium undergo the meiosis division, then there is a formation of the four haploid nuclei. Out of these four haploid nuclei in the young basidium, that is these are the two are positively strained, and the remaining two nuclei these are of the positively these are of the negatively strained. Okay, then at the distal end of the bastidium the four peg like outgrowths are formed. Here you can see that is the four outgrowths are formed on the young basidium. These outgrowths are known as the sterigmata. Okay, these outgrowths these are said to be the sterigmata. sterigmata. The sterigmata swells at their tips, and each forms the single basidiospore. Okay, here you can see that is the each of the sterigmata it will swell further, and then it will get developed into the basidiospore. Thus the four monokaryotic haploid basidiospores these are formed in the young basidium. Okay, thus the four basidiospores these are formed on the young basidium. The young basidiospore it is unpigmented, but it develops the brown or the black pigments at the maturity. At the junction of the basidiospores and the sterigma there is a small at the junction of the basidiospore and the sterigma, there is a presence of a small projection, and that small projection it is known as the hilar appendices. Okay, HILARAPPENDCENDICS. Okay, there is a small projection which is present at the junction of the basidiospore and the sterigmata, and it is said to be the hilar appendages. Due to the presence of these hilar appendages or the projections, the basidiospore it is slightly oblique at the sterigmata. Okay, then how exactly the dispersal and the germination of the basidiospores takes place? As the basidiospores mature, a drop of liquid it appears at the hilar appendages. Okay, here you can see that is the small drop of the water which is present, small drop of the liquid it is present. Okay, as the basidiospore matures, a drop of liquid appears at the hilar appendices, which remains surrounded by a limiting membrane. The basidiospore thus lies just above this drop. The drop increases in size gradually and attains a size of about one fifth of the spore. Then the basidiospore, it is suddenly shot away from the steric okay. Here you can see, due to the presence of that liquid, the size of that liquid, it gradually enlarges, and due to which suddenly that basidiospore, it get shot away from the steric. The four basidiospores of a basidium, these are dispersed in the rapid succession. When the basidiospores falls on the suitable substratum, it germinates by producing a germ tube that grows into the primary monokaryotic mycelium. Okay, all these basidiospores, these are randomly dispersed into the environment, and all these basidiospores will germinate and give rise to the monokaryotic primary mycelium. Depending upon the strain of the basidiospore, the mycelium, it may be positive or the negative strain. The primary monokaryotic mycelia forms the dikaryotic mycelium by the somatogamy. Okay, that is the sexual reproduction in the agaricus. Okay, then here you can see the presentation on the life cycle of the agaricus. Here you can see the monokaryotic myceliums, these are present in the soil. The monokaryotic mycelium, these are of the opposite strain, that is they will get fused with each other. Then there the somatogamy takes place, then diplotization takes place, dikaryotic cell is developed. In the dikaryotic cell, clamp connections are formed. Then ultimately, due to this somatogamy, there is a formation of the secondary mycelium. Then secondary mycelium twist together, and there will be the formation of a knot-like structure, and that knot-like structure, it is called as the rhizomorph. This rhizomorph then further give rise to the fruiting bodies, which are called as the basidiocarp. Basidiocarp, these are aerial portions. These are present above the soil, and this is the part which is commonly and popularly called as the mushroom. In the basidiocarp, then the basidia, these are present. That is, generally these are present on the gills, and these basidia, these are with the diploid nucleus. Okay, this diploid nucleus then further undergo the meiosis division, and there will be the formation of the four haploid monokaryotic basidiospores. Okay, of these four basidiospores, the two basidiospores, these are of positively strained, and the remaining two basidiospores are the negatively strained. Positively strained basidiospores, when they get the favorable environmental condition, they will germinate into the into the positively strained monokaryotic mycelium, and the negatively strained basidiospores, when get the favorable environmental condition, will get developed, that is they will germinate and will get developed into the negatively strained monokaryotic primary mycelium. Okay, that is the life cycle pattern in the agaricus. That is all about the life cycle of the agaricus.